Chapter 5, Fundamentals of Human Energy Transfer. The objectives are describing the first law of thermodynamics related to energy balance and biological work, define the terms potential energy and kinetic energy, and give examples of each, give examples of exergonic and endergonic chemical processes within the body, and indicate their importance. State the second law of thermodynamics and give a practical application. Identify and give examples of three forms of biological work. Discuss the role of enzymes and coenzymes in bioenergetics. Identify the high energy phosphates. Discuss their contributions in powering biological work. Outline the process of electron transport oxidative phosphorylation. Explain oxygen's role in energy metabolism. Describe how anaerobic energy release occurs in cells. Describe lactate formation during progressively increasing exercise intensity. Outline the general pathways of the citric acid cycle during macronutrient catabolism. Contrast ATP yield from carbohydrate, fat, and protein catabolism. And explain the statement, fats burn in the carbohydrate flame, why that takes place. So energy is the capacity to do work. The body's capacity to extract energy from food, nutrients, and transfer it into the contractile elements of skeletal muscle determines our capacity to move. Energy transfer occurs through thousands of complex chemical reactions that require the proper mixture of macro and micronutrients continually filled by oxygen. The term energy suggests a dynamic state related to change. Thus, the presence of energy emerges only when change occurs. First law of thermodynamics is that energy can't be created nor destroyed, but transformed from one form to another without being depleted. Conservation of energy in the body. Chemical energy stored with the bonds of macronutrients doesn't immediately dissipate as heat during energy metabolism. Large portion remains as chemical energy with which the musculoskeletal system then changes into mechanical energy. Potential and kinetic energies constitute the total energy of the system. Released potential energy transforms the basic ingredient into kinetic energy of motion. In some cases, bound energy in one substance directly transfers to other substances to increase the potential energy. They provide the required energy for the body's chemical work of biosynthesis. First of all, biosynthesis, by the way, is where the specific building block atoms of carbon, hydrogen, oxygen. Biosynthesis is the process of where the specific building blocks of atoms of carbon, hydrogen, oxygen, nitrogen become activated and join other atoms and molecules to synthesize important biological compounds and tissues. Some of these are adenosine triphosphate, which is ATP, and phosphocreatine, which is PCR, which are the main source of the cell's energy requirements. You have potential and kinetic energy. So potential energy is like the rock at the top of the hill there. It's ready to fall. Kinetic is moving, and then heat energy is what's lost or dissipated. So potential energy dissipates the kinetic energy. As the water flows down the hill, work results from harnessing potential energy, and then there's a lower potential energy at the bottom. Exergonic and endergonic reactions. An exergonic reaction is any physical or chemical process that releases energy in its surroundings. So this represents a downhill process, and it produces a decline in free energy. Endergonic reactions are chemical processes that store and absorb energy, so they take in energy, represent an uphill process, and proceed with an increase in free energy for biological work. Second law of thermodynamics um, is that the tendency of potential energy to convert to kinetic energy of motion with a lower capacity for work. So therefore, ultimately, all the potential energy in the system degrades to an unusable form of kinetic or heat energy. So kinetic is energy in motion, or heat energy, which basically is dissipated. Different forms of energy. There's chemical, mechanical, heat, light, electric, and nuclear. So interconversions of 6 forms of energy. You have sunlight energy that you have sunlight, you have light energy of the sun, which the plants take in the sun, okay, and what happens with that is photosynthesis takes place, where the chloroplast absorbs the radiant or solar energy to synthesize, and the chloroplast is in the plant. They end up taking in the solar energy, like you see there with the sun, and then it synthesizes glucose from carbon dioxide and water. So it takes in carbon dioxide and water. So then it ends up creating glucose and oxygen. So you see that there, which is that H2O and CO2 create glucose, and also oxygen comes out of the process. So that's why it's so essential that you have plants and trees and things like that. Nuclear energy is another form of energy that's created by breaking down. Basically, you have atoms reacting. Heat energy, solar panels, mechanical energy, example, hydroelectric generating plant, electrical energy there. So it talks about all of them there. Light energy, chemical energy, fossil fuel, oil burner. Okay. Forms of biological work. Or mechanical work, which is muscle contraction, chemical work, synthesis of cellular molecules. Okay, so this would be like the building up or breaking down. So synthesizing, you get proteins from amino acids, things like that. Transport work, concentration of various substances in the intracellular and extracellular fluids. Exergonic process of cellular respiration. So you have an endergonic reaction that takes in energy, which is photosynthesis, where plants make glucose, and then you have cellular respiration, which is an exergonic process, where what shows here reversive photosynthesis, where you have glucose and oxygen yield carbon dioxide and water plus ATP, and ATP is adenosine triphosphate, which enables the body to do mechanical work, chemical work, and transport work. Okay, chemical work, you see glucose, ATP. With ATP forms glycogen, which is stored in muscle and liver. You have glycerol, fatty acids. That creates triacylglycerol, which are fats, lipids. And then you have amino acids that, when you add ATP, they build protein. So this is the building up through exergonic processes. Transport work, that's a cell that you see there. And when it takes ATP basically, when you have, so triphosphates, three phosphates, when it's broken down to ADP, so it releases a high energy phosphate, and then that will take one of those key elements across the cell membrane. Okay, whether it be potassium, sodium. Factors affecting bioenergetics. First of all, bioenergetics is a field of biochemistry that deals with energy that flows through living systems. All right. You have enzymes, which are proteins that are highly specific and large. Okay, so they're protein catalysts that accelerate the forward and reverse rates of chemical reactions within the body without being consumed or changed in their reaction. Reaction rates, the operation of the rate of enzymes, the mode of action, how an enzyme interacts with its specific substrate, and coenzymes, complex non-protein that facilitates enzyme action by binding to the substrate with its specific enzyme. So that's how enzymes work. Speed up reactions, change the mode, or become coenzymes that then cause something to take place. An example of that is on page 145, which talks about how you have a substrate and then you have an active site, and then it talks about what an enzyme does in regards to that interaction. Enzymes, basically, the pH has to be correct, the temperature as well, for enzymes to actually work properly. It just depends. Enzymes have at the end of them, they have ASE, so A's is something they typically have. So like hydrolases, or lipase is something that adds water to break something apart. So you have ATP here, and it's the energy currency of a cell, adenosine triphosphate. And this is like a battery that's completely stored. When you break off that phosphate at the end there on the far right, when you break the bond, basically what it does is it releases energy. So when you're building, for instance, proteins from amino acids, it's going to use one of these phosphates to do that. Energy currency, powering biological work. So ATP is energy currency. It powers biological work. It causes glandular secretion, okay, from the hypothalamus. It causes digestion to take place, muscle contractions, nerve transmissions, circulation, heart. And like I showed you there, amino acids basically are building up to form proteins. That's an anabolic type of thing. So the interesting thing is that many processes take place in the body that use ATP. So it's extremely important that we have these things. This compound. Limited quantity of ATP. So the body stores only about 80 to 100 grams of ATP. So it provides enough intramuscular stored energy for several seconds of explosive all-out exercise. So the first energy system that is used is something called the ATP system, which is the ATP that's in your, for example, muscle, where you can, if you're doing like a vertical jump, something of that nature, it's up to about 8 seconds. And then there's another system that takes the place of that after 8 seconds, because ATP is very limited in the body. Okay. ATP represents an additional advantage due to its molecular heaviness. So ATP is heavy. That's why it's not efficient for the body to store much of it, okay, in its tissues. And because cells store only a small quantity of ATP, it must be resynthesized continually at its rate of use. The phosphocreatine system, okay, energy system. All right. So this is another system in the field of bioenergetics, okay, where the energy is flowing through a system. And the phosphocreatine system, or PCR, provides some energy for ATP resynthesis, and it releases a large amount of energy when the bonds split between the creatine and the phosphate molecules. Hydrolysis of PCR begins at the onset of intense exercise, doesn't require oxygen, and reaches max in about 8 to 12 seconds. So after the ATP system reaches its peak around 8 seconds, its transfer, the energy system that we are using in our body transfers over to the PCR energy system, which is about 8 to 12 seconds. So that would be almost like a 100-meter run. Okay. Now this is all still happening without the required oxygen. As you do a 100-yard sprint, you don't necessarily have to start breathing heavy until after that. That's why, and you're really just trying to catch up with the oxygen lost. So cells store PCR in considerable large quantities, much more PCR in cells than ATP. Okay, and it's not, the molecular weight's less. ATP and PCR, anaerobic sources of phosphate. Anaerobic means without oxygen, without the presence of oxygen. ATP is still created, okay? It's created through ATP, the use, it's created by PCR. But this is how, so basically biological work can take place by the use of these sources of energy. So you have the different bonds that release energy. So you have ATPase, which breaks the bond of the high energy phosphate to be able to do biological work. Then you have the phosphocreatine plus ADP. ADP is the uncharged battery, and then you have creatine kinase, which then creates the CR plus ATP. Okay, the creatine plus ATP. So both forms of these, when you have this bond energy, it's able to provide energy to our cells. Phosphorylation, the energy transfer through phosphate bonds of ATP to other compounds to raise them to a higher activation level. So the phosphorylation, the free phosphate, is what causes, the energy to take place. Okay. Oxidation. This is the biological burning of macronutrients in the body for energy needed. For phosphorylation, it occurs in the inner lining of the mitochondrial membranes. Cellular oxidation, if you look at it on page 150, so it occurs in the presence of oxygen, and in the cell it's occurring in the Krebs cycle, or the CAC, citric acid cycle. What it does is it involves transferring the electrons from NAD and FADH2 to molecular oxygen, which releases and transfers chemical energy to combine ATP from ADP plus a phosphate ion. During aerobic resynthesis, ATP resynthesis, oxygen combines with hydrogen to form water. Okay. So more than 90% of ATP synthesis takes place in the respiratory chain by oxidative reactions coupled with phosphorylation. A lot of the ATP the oxidative reactions is what creates most of the ATP. the quick burst of 8 seconds of a sprint up to 8 to 12 that comes from the ATP in the muscle. And then also the phosphocreatine, which is the 8 to 12 seconds, energy comes from that. It's relatively small amounts of ATP that's produced. 90% of the ATP synthesis taking place in the respiratory chain comes from the mitochondria, okay, and the citric acid cycle, Krebs cycle, same thing. Examples of harnessing potential energy. What this basically is saying is that these high energy, this electric transport chain is releasing ATP, and it's because it's the burning of macronutrients takes place and it releases electrons. Okay, that's really what's going on here. All right. You can also gain electrons where there's an exception, except the electrons are accepted, which when that happens, basically the reduction reactions are taking place. And that's how that really is in essence how cellular oxidation reduction constitutes the mechanism for energy metabolism. So you're either breaking down the macronutrients, carbs, fats, and proteins, or you're storing them. When you're storing them, that's when you're gaining electrons. So that's what this says. Oil rigs, basically. So you have oxidation, involves loss of electrons, which releases energy, and then you have reduction, involves gaining electrons, which is building up energy. It's the easiest way to say it. Macronutrient fuel sources for oxidation. So triacylglycerol and glycogen molecules stored within muscle cells, blood glucose, fat, free fatty acids, intramuscular and liver-derived carbon skeletons of amino acids, anaerobic reactions in the cytosol. So that's like where glycolysis takes place. And that's in the initial phase of glucose or glycogen breakdown, which doesn't require oxygen. And then phosphorylation of ATP by PCR under enzymatic control by creatine kinase and adenylate kinase. So that's when phosphorylation is, there's the ADP becomes ATP, okay, by that phosphate rejoining a free phosphate. So it basically is like recharging that battery. So macronutrients, the breakdown of them, whether it be fats, proteins, carbs, when they're oxidized, they release energy. Okay, so you have liver glycogen. Okay, it becomes glucose. You have deaminated amino acids. They enter the bloodstream as well. In muscle tissue, you have the energy stores in muscle tissue. We already talked about. You have ATP in there, phosphocreatine. You have triacylglycerols, which are fats. You have glycogen, which is the body's endogenous production of glucose. You have carbon skeletons from amino acids. You also have adipose tissue that is broken down, okay, to form triacylglycerols and fatty acids. These are used in the citric acid cycle, okay, in the mitochondria, or the Krebs cycle, in an electron transport chain, so that electrons are released and ATP is created. Carbohydrate energy release. Carbs are the primary functional supply energy for cellular work. They represent the only macronutrient that generates ATP aerobically. The complete breakdown of 1 mole of glucose to carbon dioxide and water yields a max of 686 kilocalories of chemical free energy. So oxidation of 1 glucose molecule in skeletal muscle yields a total of 32 ATP molecules, okay? and that's assuming there is oxygen present. That's one of the reasons why you need carbohydrates for anaerobic vigorous activity, because that's the primary energy source of that. Anaerobic versus aerobic. Glycolysis is carbohydrate breakdown. Aerobic is relatively slow process resulting in a lot of ATP, but it takes time to get to that point. Second stage of carbohydrate breakdown converts pyruvate to acetyl-CoA, which then progresses through the citric acid cycle. Then you have the anaerobic system, which is rapid but limited ATP production that produces lactate as an end product. So if there is no oxygen present, then the end product becomes lactate. All right, not pyruvate. And it's limited amount of energy coming out of it. Okay. So that's one thing to think about through glycolysis, is that the net ATP production is very small, but it happens very fast. The ability to gain energy from this system. Okay. So you have in glycolysis, this is what happens, the use of energy from about 12 seconds to about 90 seconds, where you're able to use glucose for energy source. And what's taking place here is that this is the energy source, the energy system that you're using between about 12 seconds and 90 seconds is anaerobic glycolysis. Okay. So lactate formation occurs at the end of this process. Lactate, it's different than lactic acid, because lactic acid is formed during aerobic glycolysis. It forms hydrogen ions, which is completely different than lactate, which is the end product without oxygen of the breakdown of glucose. Net yield of 32 ATP. So if there is oxygen, the end product is not lactate, but it's pyruvate, which then goes on to the citric acid cycle, which then creates more ATP in the presence of oxygen, and that's how it occurs in the mitochondria. Fat energy release. Stored fat's the most plentiful source of potential energy. 3 specific energy sources for fat catabolism. You have triacylglycerols stored directly within the muscle fiber in close proximity to the mitochondria. Circulating triacylglycerols in the lipoprotein complexes that become hydrolyzed on the surface of the tissue's capillary endothelium. Adipose tissue that provides circulating free fatty acids mobilized from triacylglycerols in adipose tissue. The breakdown of a triacylglycerol molecule yields about 460 molecules of ATP. So it's much different with fat release, right? Fat energy release than it is with glycolysis. So glucose breakdown is about 32 ATP, 460 for fat, which is much greater. It's just you don't get that energy as quickly. Breakdown of glycerol and fatty acids. Okay, so this is fats here. So you have glycerol and fatty acids, three fatty acids, really what it is. Okay, and when you have this, what happens is the glucose goes into the citric acid cycle, and then the triacylglycerol is the it's not able to basically be converted into the source that goes inside of there, because basically you have the the fatty acids, there's the two parts of the fats are the energy is broken down differently. The compound's broken down differently. Okay, that's basically the easiest way to say it. So the three fatty acids go through the beta oxidation phase, and then the glucose goes down through the citric acid cycle. Okay. So you have the glycerol. If you see there at the source pathway, the glycerol basic. So it's glycerol glycolysis and citric acid cycle is how the glycerol where it goes, and that's that part of the fat. And then the three molecules of the fatty acids end up going through the pathway of beta oxidation, citric acid cycle, and they yield a lot more of the ATP. So the fatty acids do go through the citric acid cycle, but they basically become beta oxidized. Fats burn in a carbohydrate flame. So when you deplete carbohydrates, it decreases pyruvate production during glycolysis, which further reduces the citric acid cycle intermediates, slowing down the citric acid cycle activity. So when that happens, fats aren't able to be degraded, okay, because they depend on the oxaloacetate's availability combining with the acetyl-CoA during the beta oxidation. So when the carb levels decrease, the oxaloacetate level becomes inadequate, and it reduces the fat catabolism. So fats can't be broken down as readily because they need the carbohydrates to be able to do this, because the oxaloacetate combines with the acetyl-CoA. And if that doesn't happen, then it reduces the fat catabolism. Protein energy release. Proteins can serve as energy substrates. When the deamination removes the nitrogen from amino acid, remaining carbon skeleton can enter a metabolic pathway to produce ATP aerobically. That's because the deamination it causes the protein, the amino acid, to then be transferred into a different macronutrient. Okay, so the deamination, okay, enables that carbon skeleton to go through the metabolic pathway to produce ATP aerobically. Protein acts as an energy substrate during long duration endurance type activities. So people say, well, what is protein used for? Okay, if I eat protein rather than carbs before an activity, okay, it's good for long duration endurance type activities. It's not going to give you that quick burst of energy through an anaerobic source, like such as resistance training or a quick sprint. Okay. Protein to energy pathway. So you have the amino acids. You lose that NH2, and it becomes glucose. And this is basically the deamination. So it can become glucose, which becomes pyruvate, releasing energy. The CoA, carbon dioxide is released, acetyl-CoA is there. It can go into the citric acid cycle and then create energy as well, just as the carbs and the fats did. Okay, that's all this is saying. Metabolic mill, so fats, carbs, and proteins. Okay, you have fats here, which we talked about all these already, but this is just a chart. And it just says here that the fats are fatty acids and glycerol. The fatty acids in the fats going through the beta oxidation pathway, which then end up becoming acetyl-CoA, and then that's what we talked about. Oh, we need carbs to basically create the ATPs from fats, and then the glycerol portion can actually go through the pathway of glycolysis and go directly through the citric acid cycle. And then proteins. You have proteins, which are amino acids. Deamination occurs, which we showed in the last slide or two that talked about how uh, you have um, the amine group. You have the ammonia. You have basically the ammonia comes out of it, becomes urea, becomes urine, and then that's because of that deamination. The protein becomes another macronutrient that can then be used and go into the citric acid cycle. That's why it says at the bottom. Can carbohydrates form glucose? Yes, obviously. Can they form amino acids? Yes. Okay. Uh, addition of nitrogen yields non-essential amino acids. Can they form fat? Yes, they're converted to fat. Can fats form glucose? No, fatty acids can't, but the glycerol can. Okay. Can fats form amino acids? No. Can they form fat? Obviously, yeah. Proteins? Yes. Can they form glucose? Yes. Glucogenic amino acids. And the reason they can form glucose is basically what is because of what we talked about, how they end up losing the NH2. All right, and through the deamination, and then they can form obviously amino acids because they're made up of those, and then proteins can form fat. Yes, ketogenic amino acids, and again, it's through the deamination process.